Today we'll discuss about the biological fixation of nitrogen. So first of all, what is nitrogen? It's a mineral. It's a mineral that is available in the soil in different ways. So today it's a biological fixation of nitrogen. And so we know that urea is a good source of nitrogen, but it is a fertilizer. In the soil there are many organisms, microorganisms that help the plant to utilize the nitrogen and will develop their NH3 product. So for example, here N2 will be converted to NH3. It is called ammonia. It's a final source which used by plant. But here this N2 is produced how, where and how it is utilized. We have to know that. For example, microorganisms like free-living bacteria like Bacillus anabina. Symbiotic relations will be shown by the plants like peas and the plants like rhizome, like ginger, etc. For example, the free-living bacteria we know that is Rhizobium leguminosarum. So it is available in the legume plants like pea. Rhizobium is a symbiotic bacteria that was present in the legume plants. So here it is an aerobic organism. It is also anaerobic during the N2 fixation, because if this bacteria is aerobic, it can't generate the nitrogenase enzyme that helps a plant to absorb the nitrogen and complete the nitrogen cycle per atom. Nitrogenase is an enzyme that produce both, that have a protein structure along with molybdenum and ferrous. First step is nodule formation, and second step is N2 fixation, and third step is nitrification, and fourth one ammonification. Nodule formation are nothing but globular structures that develop on the root hairs, or the primary root hairs. Here in the first step we discussed the nodule formation. The curling of the root hair is called shepherd's screw. Two types of useful things here will happen. First thing, it helps the N2 fixing cells to active more, and the second thing, the bacteria will not allow another harmful bacteria to attack the root. This is a symbiotic relationship between the root hair and the bacteria. N2 fixation. Conversion of N2 to NH3 is called N2 fixation. And now for example, it's a nitrogenous enzyme. This is called the binding site. Will be cleaved by the attacking of N2. That supplements that the plant absorb the N2. This nitrogenous enzyme will form a binding with the nitrogen. First, it's a binding substrate. So next, here if you check, no molecule will have, for example, nitrogen is a triple bond molecule we know. So we have to adjust the enzyme reaction in a particular way. So here it will undergo the reduction reaction. So these many reduction reactions will take place because the plant must not require the oxygen, and the legume hemoglobin that was present in the particular bacteria will help the oxygen to remove at that particular stage. So in the last we can find here, in the last step we can see NH3 plus NH3. Two NH3s are developed, and the nitrogenous enzyme will be set free such that it can also act one more time. In this way, the entire N2 fixation takes place biologically. So what is nitrification? NH3 will be converted to NH4 ions. So again, nitrification will take place by the bacteria called Nitromonas and Nitrococcus. NH4 plus will react with the oxygen and form NO2 minus plus H2O. This is called nitrate and it is water. This was carried out with the help of nitrosomonas Nitrosomonas and Nitrococcus. So these type of bacterial organisms will mainly adjust the plant to convert the NH3 to NH4, that is ammonium to ammonium ions. NO2 minus plus oxygen gives rise to NO3, that is the nitrates plus water. It is run by nitrobacter. So in this way, biological fixation takes place. Thank you. Takes place. Thank you.